Welcome friends, I am Dr. Dinesh Kumar Mishra, Professor of Pharmaceutics, College of Pharmacy, IPS Academy, Indore. I am back with another episode on Pharmaceutical Aerosols Part 2. In this episode, we are going to continue our previous talk. In the previous talk, we have talked about the basics of aerosols, components of aerosols, propellants, general formulation and manufacturing of aerosols. In this talk, we will talk about packaging of aerosol preparations, evaluation methods and their pharmaceutical applications. So friends, let's begin our today's episode. As we know that pharmaceutical aerosols are a type of pulmonary drug delivery system and considered as one of the best approach in comparison to other mode of administration. They are similar to other dosage forms and require similar considerations with respect to formulation, product stability and efficacy. However, they differs in terms of container function, valve assembly and an added component. Its merits over the other route of administration augment its wide range of applicability in the treatment of illness including asthma, chronic obstructive pulmonary diseases. Some of its benefits include the possibility of direct targeting the drug to the site of action, avoidance of first pass metabolism, quick onset of action and systemic side effects reduction. Hence, aerosols could be very useful delivery system. Packaging of aerosol preparations. Quality assurance comprises an important concept of the total approach to good manufacturing practice. To ensure high quality of drugs, the quality management system must take into consideration the requirements of the local authorities and the legislation, the product, the production process and the policy of the manufacturer. The foremost requirement for packaging must be a good design as relevant to the need of the product, product pack stability, compatibility, manufacturing and distribution system, the patient and the legislation. Successful packaging of pharmaceutical aerosols necessitates special knowledge, skills and equipment. Like other pharmaceutical products, this operation must be performed under strict supervision and adherence to rigid quality controls. A unique aspect of pharmaceutical aerosol compared to other dosage forms is that the product is actually packaged as part of the manufacturing process. In other dosage forms, mostly product is manufactured and then packaged in suitable container. Two methods have been used to package aerosol products. Unlike non-aerosol products, part of the manufacturing of necessity takes place during the filling operation. The propellant and product concentrate must be brought together in such a way as to ensure uniformity of the product. Depending on the nature of the product concentrate, the aerosol can be filled by a cold filling or a pressure filling process. As we have already discussed these processes in our previous talk under manufacturing of pharmaceutical aerosols, hence here we are not going to repeat those filling procedures. Briefly, in cold filling method, temperature of the concentrate is lowered below room temperature, that is minus 30 degrees centigrade to minus 60 degrees centigrade, followed by addition of cold concentrate to chilled container. Then propellant is added and valve is clamped in place. Finally, the container is passed into a butterbath for ensuring no leakage or distortion in the container. While in pressure filling method, concentrate may be chilled for reducing vaporization of any volatile solvent or propellant. Then concentrate is added to an open container and valve is clamped in place. Further, propellant is added under pressure through the valve. Finally, the filled container is passed through the water bath. Usually, aerosols products have a protective cap or covering that fits snugly over the valve and mounting cup. It protects the valve against contamination with dust and dirt. Cap is generally made up of plastic or metal and used for decorative function also. Now we will talk about labeling and storage. Aerosols preparations that are to be dispensed only upon prescription are usually labeled by the manufacturer with plastic peel away labels or easily removed paper labels so that the pharmacist may easily replace the manufacturer's label with this label containing the directions for use specified by the physician. Mostly other types of aerosols have the manufacturer's label printed directly on the container or on firmly affixed paper. Additionally, apart from usually labeling requirements for pharmaceutical products, aerosols have specific requirements for use and storage. From safety point of view, warning must be mentioned on the label for users not to puncture pressurized containers, not to use or store them near heat or an open flame, and not to incinerate them upon exposure to high temperatures, and they must get burst an aerosol container. Quality control for pharmaceutical aerosols. Generally, there is no basic difference between methods used for the preparation of pharmaceutical aerosols and non-pharmaceutical aerosols, but there are different standards and specifications for their production. Hence, quality control testing of pharmaceutical aerosols has to be done. That includes propellant testing, testing of valves, actuators and dip tubes, container testing, weight checking, leak testing. and spray testing also. Now start with first one, that is propellant testing. In propellants which are used for medicinal and pharmaceutical aerosols need special handling and specific test procedures. Vapor pressure is usually determined and compared with specification. Further, testing can also be done by density determination. Gas chromatography and IR spectroscopy can be used for propellant identification. Purity and acceptability of propellant is checked by moisture, halogen and non-volatile residue determinations. Valves, actuators and diffuse testing. Sampling is done as per military standard, that is MIL STD 105D, a suitable test method that was developed for meter dose aerosol by Aerosol Specification Committee, Industrial Pharmaceutical Technology Section and Academy of Pharmaceutical Sciences. The objective of this test is to determine the magnitude of valve delivery and degree of uniformity between individual valves. Standard test solutions were proposed to rule out variations in valve delivery. Standard test solutions were proposed to rule out variations in valve delivery. For testing this, there are table is showing the test solution of different composition. Now move toward testing procedure. In this, initially 25 valves are selected and placed onto suitable containers. These containers then filled with specific test solution. An actuator having 0.020 inch or larger unrestricted orifice is attached to the valves. The filled containers are placed in a suitable atmosphere at a temperature of 25 degrees centigrade. When the product attains this temperature, the valve is actuated to full extent for nearly 2 seconds for complete dispensing of contents in a single delivery. This procedure is repeated for a total of 2 individual deliveries from each 25 test units. The content delivery through valve per actuation in microliter is equal to individual valve product delivery weight divided by specific gravity of test solution. Now what are the limits for this valve acceptance? The limit is, if the delivery is 54 microliter or less, the limit is plus minus 15%, and if it is 55 to 200, the limit is plus minus 10%. Now move towards if the delivery is for 50. For 50 deliveries, valves can be rejected if 4 or more deliveries are out of the limits. Another 25 valves are tested if 3 or more deliveries are out of the limits. Whole lot is rejected if more than one delivery is outside specification. Another 25 valves are tested when 2 deliveries from one valve are beyond limits. Lot is accepted if not more than one delivery is outside specifications. Now what about container testing? Containers are tested as per standard sampling procedures. Uncoated as well as coated metal containers need to be examined for lining defects. Quality control characteristics include degree of conductivity of an electric current as a measure of the exposed metal. Glass container must be checked for flaws. Weight checking testing. Weight checking testing that is carried out by periodically addition to the filling line tiered empty aerosol containers which after being filled with concentrate then removed and weighed accurately. The same procedure is applied for weight checking after propellant addition. Last one is leak testing. Leak testing, it is a means of valve crimp checking and detection of defective containers due to leakage. This is done by crimp dimension measurement and comparison with specifications. Valve closure efficiency is finally tested by passing the filled containers through the water bath. Another testing is spray testing. Most pharmaceutical aerosols are 100% spray tested. It serves to clear the dip tube of pure propellant and pure concentrate. It is also used for checking of the defects in the valve and the spray pattern. Before moving towards module 4, that is evaluation method of aerosols, I would like to suggest you please visit our website, that is www.cec.nic.in for the reading and learning and more topics associated with your B.Pharm syllabus. Here you will find many lectures, e-contents, LORs and many more. Hope this site will be catalyst of learning for you. Now that is evaluation methods. Now coming to evaluation methods of aerosols. As far as evaluation of aerosol preparation is concerned, it is done on the basis of some major parameters that are as follows. First one is flammability and combustibility, physicochemical characteristics, performance, biological characteristics, stability testing. These major parameters are tested on sub-parameters that are given below. Under the flammability and combustibility, we are going to check flash point and flame extension. Second one is physicochemical characteristics. Under this test, testing of vapor pressure, density, moisture content, propellant identification, concentrate propellant ratio. Under third one, that is performance. Under this, aerosol valve discharge rate, spray pattern, doses with metered valves, net contents, foam stability, particle size determination and leakage. And last one is biological characteristics. Under this test, testing of therapeutic activity and toxicity study. Now we'll discuss individually these evaluation parameters. Now start with first one, that is flammability and combustibility. Under this, the aerosol proportions can be determined by the following methods. Flash point. Now this flash point is determined by standard tag open cup apparatus. The aerosol formulation is chilled to a temperature about minus 25 degree Fahrenheit and transferred to test apparatus. Then test liquid is allowed to increase slowly in temperature. The temperature at which the vapors ignite is considered as flash point. Now another parameter, that is flame extension. It helps to indicate the effect of an aerosol product on the extension of an open flame. The product is sprayed nearly for 4 seconds into a flame. Extension of the flame will depend upon the nature of product. The exact length can be measured with a ruler. Now move towards another parameter, that is physicochemical characteristic. Under this test, first one is vapor pressure. That can be measured with the pressure gauge or through use of a water bath, test gauge and special equipment. This parameter is very important and its variation from container to container should be determined, since excessive variation gives indication of air existence in the head space. For accurate measurement, puncturing device can also be used. Another parameter, that is density. The density of an aerosol formulation may be accurately determined by the use of a hydrometer or a pycnometer. These methods are generally used for density of non-aerosol preparations. With slight modifications, these methods can be used for aerosol preparations. A pressure tube is fitted with metal flange and a hook valve that allow introduction of liquids under pressure. The hydrometer that is placed inside the glass pressure tube. Adequate sample is placed through the valve to cause the hydrometer to rise halfway up the length of the tube, and directly density can be read. Another parameter, that is moisture content. For determination of this moisture content Karl-Fischer method is widely accepted. Gas chromatography can also be used for this purpose. Another parameter, that is propellant identification. Generally, gas chromatography and IR spectrophotometry can be used for identification of propellant and also for indication of proportion of each components in a blend. Another parameter, that is performance. How performance is checked? Aerosol wall discharge rate is first parameter that can be determined by taking known amount of an aerosol formulation and discharge of contents for a specific period of time using standard apparatus. After time limit, the container can be reweighed and change in weight per time dispensed can be recorded as the discharge rate. This discharge rate can be expressed as gram per second. Another parameter is spray patterns. Method for determining spray pattern is based on the impingement of the spray on a piece of paper that has been treated with a dye talc mixture. According to the nature of the aerosol formulation, oil soluble or water soluble dye that is preferred. The particles that strike the paper cause the dye to go into solution and to be absorbed on the paper. This gives a record of the spray which can be used for comparison purposes. The amount of material coming into contact with the paper can be controlled by attachment of paper to a rotating disc that is having an adjustable slit. Another parameter, that is doses with metered valves. This parameter requires consideration of number of points. First one is reproducibility of doses each time the valve is depressed. Second one is amount of medication actually received by the patient. Doses reproducibility may be determined by assay techniques, whereby 1 or 2 doses are dispensed into a solvent onto a material that absorbs the active ingredients. These solutions can be further assayed and the active ingredients amount is determined. In another method, accurate weighing of filled container is done, followed by dispensing of several doses. The container then can be reweighed and the difference in weight divided by the number of doses dispensed gives the average dose. It must be repeated and results can be compared. Amount of medication received by a patient determination is difficult procedure because all of the dispensed material is not carried to the respiratory tract. An artificial respiratory system can be used for this purpose, and the parameter is net contents. Numerous methods can be used to ensure that sufficient product has been placed into each container. The tiered cans that have been placed onto the filling line are reweighed and the difference in weight is considered as net contents. The other method is a descriptive method which consists of weighing a full container followed by dispensing the contents. The contents are then weighed and difference in weight gives an indication of amount present in the container. And next parameter is foam stability. The life of foam can be ranged from a few seconds to hour or depends upon the formulation. Several methods can be utilized, which includes a visual evaluation, time for a given mass to penetrate the foam, time for a given rod that is inserted into the foam to fall, and the use of rotational viscometers. Next parameter is particle size determination. Cascade impactor and light scatter decay methods, these are mainly preferred for determination of particle size. Cascade impactor, that is based on the principle in which a stream of particles projected through a series of nozzles and glass slides at high velocity. The larger particles becomes impacted fast on the lower velocity stages, and the smaller particles passed on and are collected at higher velocity stage. Light scatter decay is based on principle of settlement of aerosol under turbulent condition, and the changes in light intensity of a Tyndall beam are measured. Another important parameter for aerosol testing is biological testing. Therapeutic activity and toxicity study are considered under biological testing of aerosol products. How to check the therapeutic activity? For inhalation type of aerosols, the therapeutic activity is dependent on the particle size, while in case of topical aerosols, activity is determined by application of formulation to the test areas and amount of therapeutic active substances absorbed. Next parameter is toxicity study. This study is also considered for both inhalation as well as topical aerosols. Topical aerosols are checked for irritation or chilling effect in the skin. As such, no good test is available presently, but thermistor probes attached to the recording thermometer can be used to measure change in skin temperature for a particular period of time. Inhalation toxicity study is carried out by exposure of test animals to vapors sprayed from an aerosol container. Another important parameter is stability testing. It is one of the most important considerations in the pharmaceutical aerosol formulation. Components used to prepare these formulations must be studied for stability issue, and also the container product interaction must be considered. Leaching of extracts from container can cause severe harm. They must be identified and rectified. Similar consideration must be given to valve components also, as varieties of materials are applied for manufacturing of container, valve and dip tube. It is very difficult to ensure the interaction between the materials and the drug. Stability testing of aerosol must consider following area. First one is concentrate and propellant, next one is container, another one is valve. Pharmaceutical applications of aerosols. Numerous therapeutic agents has been administered or applied to the body by means of the aerosol dosage form. The aerosols in the field of pharmacy can be used for local as well as systemic delivery of the drugs. First one is local delivery. For local treatment, aerosols can be applied topically to the skin and a uniform thin layer without touching the affected area. They can also be used for diagnosis purpose, prevention or control of lung disease. Over the last 10 years, the importance of inhalation therapy for asthma has increased, with virtually all asthmatics now using aerosolized medication for the treatment of their asthma, since adequate therapeutic concentration of drug is achieved in the respiratory tract with the aerosol. Now, next one is systemic delivery. The recent developments of a new class of drugs have generated greater interest for alternative administration to the enteral route because of its proteolytic activity and the catabolism of the first pass effect. In contrast to oral delivery, aerosols were found to be effective in systemic delivery, gene therapy and vaccination. Aerosol preparations have diverse applications and can be applied by various routes of administration. It includes topical, oral, rectal, vaginal, nasal, inhalation, etc. Topical aerosols. The topical aerosols can be applied in a convenient manner on affected area. The formulation can be applied to the desired surface area without the use of the fingertips like other types of topical preparations. Benefits of these types of formulations include convenient to use, no need to touch skin, minimal contamination of unused product, convenient aerosol package for use on the skin include the anti-infective agents like povidone iodine, tolnafit, thiomersal, adrenocorticosteroids like betamethasone, dipropionate, valrate, dexamethasone, triamcinolone acetonide, and the local anesthetic dibucaine hydrochloride, benzocaine, antifungal sprays, miconazole, vaginal and rectal aerosols. Aerosols used as foams for intravaginal delivery similar to creams. Commercially available aerosol foams contain estrogenic substances and contraceptive agents. The product package contains an inserter which is filled with foam and the contents are placed in the vagina through activation of the plunger. Some commercially rectal foams use inserts. Such product is Protoform, contains Pramoxine hydrochloride to relieve inflammatory anorectal disorders. Now move towards oral aerosols. Aerosols are used to deliver the drugs to the oral cavity, generally administer the product sublingually. For example, sublingual nitroglycerin aerosol formulation is sprayed under the tongue and it delivers 0.4 mg of nitroglycerin per actuation, depending upon the valve used. Now, inhalation aerosols or nasal aerosols. Inhalation aerosols deliver the drug directly to the nasal mucosa of nose. The most of nasal aerosols contain steroids which are used to treat sneezing, nasal congestion and running nose along with hay fever, rhinitis and allergies. Examples for inhalation and nasal aerosol contain steroids beclomethasone dipropionate, dexamethasone, sodium phosphate, triamcinolone acetonide and budesonide, nasal decongestants, salbutamol, albuterol sulfate, sodium chromoglycate, fluticasone propionate, nedochromyl sodium, etc. Now friends, it's time to summarize our talk. Aerosols are like other dosage forms and necessitate analogous considerations with respect to formulation, product stability and efficacy. Packaging of pharmaceutical aerosols demands special knowledge, skills and equipments. This operation must be done under critical supervision and stick to inflexible quality controls. Quality control testing of these preparations include testing of propellants, valves, actuators, diff tubes, container, weight checking, leak testing and spray testing. Aerosolic preparations are evaluated on the basis of certain major parameters that are flammability and combustibility, physicochemical characteristics, performance evaluation, biological testing and stability testing. Numerous therapeutic agents has been administered or applied to the body by means of aerosol dosage forms for local as well as systemic delivery. These preparations can be applied by various means: topical, oral, rectal, vaginal, nasal inhalations. In summary, this module will help students to understand about packaging requirement of aerosols, quality control testing, evaluation of aerosols, therapeutic applications of aerosols. Thank you.